Everyone's heard of the Ice Bucket Challenge. Inviting your friends to dump ice-cold water on their heads, or else donate to the ALS Foundation. Luckily for the foundation, many generous people not only chose to dump ice, but also dumped their money into the foundation, which raised over $100 million. But not everyone knows what ALS actually is and how terrible of a disease it can be. ALS, or amyotrophic lateral sclerosis, is a neurological disease caused by the breakdown of motor neurons in the brain and spinal cord. Normally, the brain sends signals to muscles via motor neurons, causing the muscles to contract. With ALS, the neurons begin to break down and lose their ability to transmit signals. These incomplete signals cause spastic muscle movements, an early indication of ALS. Over time, the neurons lose their ability to transmit any signals at all. Once the neurons are no longer sending signals to muscles, the inactive muscles begin to shrink, or atrophy, and lose their strength. Most people are confined to a wheelchair with limited mobility for the rest of their lives. Unfortunately, there is presently no cure for ALS, and the only real treatment is a drug that only extends life by 2 to 3 months, and doesn't necessarily improve the quality of life during that time. So, you may ask, what can we do to help those with ALS? With the discovery of stem cells, be they embryonic, induced pluripotent, or adult stem cells, scientists have started developing new treatments. You've probably heard a lot of news about the potential of stem cells, as well as the moral concerns around their use. However, for many medical treatments, including those for ALS, the stem cells being used are typically collected from the people themselves, sidestepping the ethical concerns over using certain stem cell types, such as embryonic stem cells. In recent years, scientists have instead chosen to focus on the other two main types of stem cells, adult and induced pluripotent stem cells, or IPS in short, as there are less moral concerns and greater availability. Particularly for ALS, scientists have begun studying how to use both adult stem cells and IPS cells for many reasons. First, let's look at adult stem cells. These cells can be found throughout our bodies. Their job is to replace old or damaged cells. Since they are found and don't have to be made artificially or taken from embryos, they are easy to isolate and reproduce compared to the other stem cell types. To maintain these populations of cells, the body has special locations called niches. These niches are the perfect environment for adult stem cells. They provide both chemical messages, which tell the cells what to do, and a physical environment which provides the right nutrients for the cells. Both of these help to maintain that population of adult stem cells in just the right way, allowing the stem cells to keep their stemness, meaning that they will live in a dormant state for an extended period of time. This niche environment also provides signals, like letters or information in the mail, to the stem cell, and they tell the stem cell what to do and what to become. When the stem cell receives the right signal, it undergoes a process called asymmetric division, which produces 2 daughter cells. One of these cells is a copy of the stem cell itself, and the other is the cell that will differentiate into a specific cell type, and then move to the location where it is needed. One benefit of using adult stem cells is that they can be taken directly from a patient and used to treat that patient's illness. Since these cells come from the patient, this reduces the chance of rejection by the patient's immune system. This is very important when trying to develop new therapies because the risk of the treatment not working is reduced compared to other types of cell-based treatments. Most importantly, however, is that the patient will not need to take medications that reduce the immune response to these therapies, which can increase their chances of getting another sickness or disease. Using autologous, or the patient's own cells, it is also helpful in that the cells are more readily accepted into the existing biological systems where they are placed. Meanwhile, IPS cells also have this ability to minimize an immune response. IPS cells are made artificially in a lab, but they come from mature, already differentiated cells in the body. Differentiated cells from a patient are taken and placed in cell culture. These cells are then treated with viruses that tell the cells to express certain proteins that turn these cells into stem cells, sort of like a reverse aging process. These cells then gain the ability to become a completely different cell type than from before, And since these cells are taken from the patient, they also are less likely to be rejected once placed back into the patient. While it is ideal to use the patient's own cells, adult stem cells can be taken from other people without the illness and then placed inside the patient. Since the body's first move is to reject these foreign cells, doctors retrieve cells from people with similar proteins on their cells so that the patient's body is less likely to reject the new cells. This way, in genetic diseases such as ALS, once the stem cells begin to differentiate and replace the damaged cells, they won't be inclined to become degenerative like the cells they replaced. Another advantage of stem cells is that they all have some degree of potency, or rather, how many possible cell types they can differentiate into. Adult stem cells are considered multipotent, meaning that they have the ability to differentiate into many different types of cells. The other stem cell types are referred to as pluripotent, as in the case of embryonic stem cells and induced pluripotent stem cells, meaning they can become nearly any type of cell and are the most flexible of the stem cell types. So, back to the main point at hand, what researchers are really interested in is developing a safe and effective treatment. Ideally, they want a stem cell that doesn't have to go through many steps to become the cell type that they want. In the case of adult stem cells, there are less steps to go through to reach the right cell type as they are more limited. In the context of ALS and finding the right treatment, this means that an adult stem cell from the right lineage, one that becomes cell types that are found in the brain and spine, is necessary. Why is all this important? When scientists study a disease such as ALS, where the causes are at least partially genetic, it becomes important to look at what cells or biological cues are missing. In other words, it is important to understand what is actually causing the disease. In the case of ALS, the motor neurons are continually degenerated and the muscles that they are connected to start to atrophy, where they start to become weaker and smaller due to inactivity. Because the native cells that are already in the body aren't functioning correctly, it is thought that a cell-based therapy, especially with stem cells, might be able to replace the cells that are at the root of the disease. As we have been talking about stem cells and their possible applications, let's focus on some applications that are currently being tested. How stem cells are applied when treating ALS depends on the characteristics of the certain kind of stem cell being used, since all types each have their own pros and cons. Two of the most important applications are testing therapies for treating ALS using cell culture techniques, called in vitro work, and treating ALS with stem cells delivered directly into the body. First, induced pluripotent cells can be made from the grown cells of a person with ALS. Like mentioned before, iPS cells are made by taking mature cells, in this case skin cells, and treating them with viruses to turn them into stem cells. While in vitro, these cells will then divide multiple times and increase their population size. They will then be given different proteins that will induce them to become neurons. Since these cells are from patients with ALS, these lab-grown neurons will be as defective as the patient's neurons. Researchers can then test a variety of different treatments on the neurons until they find one that repairs the faulty neurons. This treatment can then be tested further in other models, and hopefully, one day it will be used to treat ALS. The second application is treating ALS directly with stem cells. Since ALS causes motor neurons to malfunction and degrade, researchers are trying to replace the neurons that aren't working with new ones that have been derived from stem cells. Adult stem cells are taken from another person's bone marrow in their hip, and the cells can be replicated in vitro. The adult stem cells are then injected into the muscle and the area around the spinal cord of the patient with ALS. These injected stem cells are then exposed to growth factors from the surrounding tissue that encourage their differentiation into motor neurons. These cells will hopefully replace the damaged neurons and restore functionality of the muscles. This approach, however, has a long way to go, and it must be further developed to make it a viable treatment. Much more needs to be done in order to stop ALS in its tracks. While just about $100 million has been donated to ALS research, money will not be the only thing needed to find a viable therapy. Bright minds and creativity will also play a crucial role in developing these therapies and will pave the way for future developments in medicine.